allows us to read and write cells, tissues, organs, ecosystems, and a lot of this, it's not limited to biotechnology, but a lot of it is focused on developmental biology, which includes both tissue diversity and aging. So we can make almost any cell type from pluripotent stem cells into any maturity level, age level, or tissue type. We also have developed ways of doing editing of both the genome and the epigenome. I could go on, but let's get on to the questions.
Sure. So you're involved with a lot of different companies. I just want to know if you have any favorites that you of the list of different genetics endeavors that you have going on.
Well, certainly there's some new ones having to do with machine learning that I think are going to be quite impactful in developing therapies. They're diagnostic methods, that the sequencing based methods that I think we could have used for infectious disease and could, as well as inherited diseases, that would save us trillions of dollars in both cases. But when you ask someone, when the genie asks you for one wish, your wish should be more wishes. And so my favorite company is Rejuvenate Bio, which is hopefully going to grant us all more time to do our job.
Do you think that aging is programmed?
Aging is definitely programmed. It doesn't mean that it's easy to reprogram it, but the mouse is going to die after 2 years, and the bowhead whale after 200, and a lot of the tissues are going to fail simultaneously, possibly due to master regulators, but possibly due to simultaneous planned obsolescence. But yeah, there's a program, and or 9 programs that interface in various ways, and we're getting to the point where we have a half-empty, half-full glass, which is a fine time to do engineering. You don't need to understand absolutely everything in order to start doing very coherent, useful things.
So one of the questions was, how far along are the genetic reprogramming methods that you are working on? It's a very, very vague. I think you're muted. Hold on. Sorry, you're muted.
Yeah, so what happened is I muted the cough and then found out that I can't unmute.
Yeah.
And then you unmuted me, but you needed me to unmute as well. So anyway, we're good now. And your question is?
Oh, sorry. How far along are the genetic reprogramming methods that you guys are working on?
So they're about, I just gave a talk the other day where there are 11 different ways, at least, of doing aging reversal. And one of the more promising ones is aging reversal with the OSKM factors that we use for doing reprogramming of pluripotent stem cells. But it's only one of 11 ways, and there are probably various combinations as well. How far along is it? Well, I mean, we've produced 3 peer-reviewed papers on this in animals, so preclinical. Some of these are going into, as gene therapy form, into dogs, for which there is a veterinary product that people will help support the further research. Veterinary FDA approval is much less expensive and faster than human, and that's part of the reason we prioritize that as well.
what is your opinion on the three main approaches to longevity: slowing metabolism, repetitive maintenance, and genetic engineering?
Well, they're
So,
not exclusive. I mean, you can use genetic engineering for the first two. I mean, it's kind of apples and oranges. But anyway, I mean, I think genetic engineering is is a good way of doing almost everything. And I would say rather than two ways, two things to which you could apply genetic engineering is more like 11. And these include, you know, getting rid of senescent cells, using the OSKM factors to do partial reprogramming towards embryo, but not all the way there. You know, telomeres, non-cell autonomous blood-borne factors, and so forth. The list goes on. These are, I focus on aging reversal rather than longevity because the clinical trials are much faster.
oh, okay. Um, so have you heard of the recent BioTime Lineage clinical trial where they saw first in human retinal regeneration with RPE stem cell therapy? And what are your thoughts on that, if you have?
Well, there are a number of successful retinal therapies. There's cell therapies, there's gene therapies, and there's editing. The first in vivo editing of any sort was done in the retinal, and it's no coincidence that all three of these cutting-edge technologies, gene therapy, cell therapy, and gene editing, are done in the retina because it's a particularly accessible, dispensable tissue. And they're all they're all looking quite good as therapies, and it's still a dramatic impact when you cure a blind child. He goes from being unable to walk without a stick to throwing a baseball from his bicycle.
That makes sense. Which category of aging seems most difficult to overcome?
Category. Well, so I think that there's kind of two major camps. There's the epigenetic camp, which is this regulation, and the other damage camp, which is this damage, and you have to go in there and kind of fix it the way you would fix a salt corroded car. So I think they both have an element of truth. I think that if you get the epigenetics right, the cells will tend to repair themselves. But there are certain kinds of damage which are very hard to reverse. So, for example, if you delete a gene, you can't just reverse that the way you can go from A to G and G back to A. You have to get that information from somewhere. And so I think those are particularly challenging. But even major loss of organs and limbs, in principle, those can be manufactured and replaced. Also, anything that involves serious damage to the brain is hard to replace because there aren't that many neuronal stem cells in a normal brain. Now, with synthetic biology, you can do anything, but you're asking, right now, at this moment, is the most difficult is the intersection of damage with cells that are not replaceable because that's where your memories are.
Do you think that the current hallmarks of aging are adequate or close to being adequate, or are we still pretty far off from really nailing down what it is how to define aging?
Well, I think the hallmarks of aging are adequate, but you have to say for what? They are adequate for going forward in engineering. I think that people who say you have to have complete understanding of all the systems biology, you have to have a complete list of molecules or modifications, ordinary differential equations, all that. I think that's obfuscating the fact that a huge fraction of engineering is trial and error on top of good theory, but the theory doesn't have to be complete. For example, smallpox vaccine, was started in the 1500s, restarted in the 1700s. It was very successful with complete ignorance of virology and immunology.
All right. Well, I think we got through a bunch of those forum questions much faster than I expected, but that's okay because that means that's more time for the audience. And so we're gonna unmute. I'm gonna allow participants to unmute themselves, and hopefully we have a controlled chaos. We have, oh, cool. So there's a hand function in Zoom. So we're gonna have Lawrence ask the first question. Everyone should be able to unmute themselves at this point.
Great.
Okay.
Yeah. All right. Hi.
Yep.
I was wondering about your startup for rejuvenating dogs. Why not choose cats? Because they're smaller, so you can have cheaper gene therapy in smaller quantities, and they're also more genetically uniform.
Yeah, well, I'm not convinced they're more genetically uniform, but they are smaller. The cost of manufacturing the gene therapy is actually fairly low. Most of the costs come from recovering the R&D costs of doing the clinical trials, especially at the beginning. Aging is nice because, in contrast to most uses of gene therapy, which are rare diseases, those you have a very small population that are going to be paying off the clinical trials, while with aging you could have a very, very large population, and hence lower costs, and hence even larger populations. Dogs and cats, I think somebody did a market survey and found that dogs were, owners paid more for end-of-life medicine, and there were more of them and more per dog, more per pet, rather. I think that's it. Also, the CSO is a very, and who was a postdoc in my lab, Noah Davidson, loves his dog. So.
Nice. All right. So YP is going to be next.
Hi. Could you talk a little bit about modern gene editing techniques and maybe recent developments to them?
Yeah, sure. The short version of that very long question answer would be they can be broken. If you broaden the topic and then shrink it down again, you broaden it. There's addition, subtraction, precise editing, and epigenetic editing. And if you so there was 3 different ways of making precise cuts, either double strand or single strand breaks, which are highly mutagenic, but they tend to make a mess, which I call genome vandalism. So that tends to be subtraction. Addition is just gene therapy or transgenics. It's quite old. You might not even consider that editing. Precise editing is deaminase, which C's, T's, and A's and G's. It's very limited, and you tend to make a cluster of changes, so it's not really as precise as you might hope. There's a so-called primer editor, which is like homologous recombination, except it's built into the machinery. It's not very efficient yet. All these things are subject to change. And then finally, there's a box that you could put 3 things in: recombinase, integrase, and transposinase. And these are probably the most sophisticated. They'll allow you to put in big pieces of DNA or small ones, and you can make any change you want. So that's the one I'm most excited about. It's the newest one to make it into a startup company, Tessera. It's not the newest one. We actually have been working on it for 20 years. So, but it's newer than CRISPR in terms of people taking it seriously.
Okay. Looks like Dylan is next.
Thank you very much for your time, Professor Church. I'm a doctoral candidate interested in the applied biology of aging, and I have, obviously a great deal of interest in your lab's work on developing gene therapies oriented towards diseases of aging. So my question is, right now, the adoption of gene therapies into medicine has been fairly limited. There's only, a handful approved to date of therapies approved to date. In your opinion, what is the biggest obstacle at this time to more widespread use of gene therapies, like from a research perspective, and which areas of research do you think are most important for overcoming these obstacles?
Right. So, in a certain sense, the gene therapies, it's a handful, but it's moving very quickly. In the same sense that there was only a handful of monoclonal antibody therapies, and they became the most rapidly advancing front in therapeutics for a while. Gene therapies have been mostly associated with rare genetic diseases, which, because of the Orphan Drug Act of 2008, anyway, the Orphan Drug Act, they're profitable, but they are expensive, and so I think. I don't think there's a particular barrier to gene therapies taking off. It'd be nice if they're applied to a large market, as is the case for age-related diseases. Aging affects almost every form of morbidity and mortality. Even falling down or having accidents are age-related. COVID is age-related. So I think that will help us get even more momentum still.
Thank you, Professor.
Okay, it looks like Jason was next.
Hi. Some of the gene therapies with adenoviruses that exist in clinical trials seems to be promising at meeting their endpoints. So what are the advantages that CRISPR gene therapies have over the current gene therapies?
So the most heavily approved vector for delivery is adeno-associated virus, which may be what you had in mind. But delivery is, in addition to delivery, there has to be a mechanism of the genetic component. Gene therapy usually refers to addition of genes that are missing, or addition of genes that would be valuable. While CRISPR is typically about subtraction. It's about removing things that are dominant interfering factors. So it's not that one is better than the other, it's they do different things. So if you have too little of something, you want to do gene therapy addition. If you have too much of something, then you want to do subtraction. If you look through the list of rare genetic diseases, I think there's a slight excess of things that need addition rather than subtraction. So CRISPR is a relative latecomer to the field, and most of the almost all of the approved gene therapies are addition rather than subtraction. CRISPR can help with addition. It can also do precise editing, but that's the exception to the rule. Mostly, it's about subtraction.
Thank you.
We got Alexander.
Hello. Thank you for all the work that you do here. My question is, when I tell people that I'm interested in the medical intervention into the aging process, they usually are either dismissive or they are sometimes outright horrified. I think it's natural, aging is natural, you shouldn't interfere with it. I imagine you get reactions like that yourself from time to time. I'm just wondering how you respond to such concerns.
Well, there's nothing, medicine is not about natural. It's specifically about intervening. I think that part of the reason we reframed it from longevity to aging reversal is that we're defining aging reversal as reversing age-related diseases. Or now it could be that if we have a drug that's sufficiently focused on the core part of aging, it will address multiple diseases at once. So you apply it to one, and the next in a preventative manner for many others, and it could result in longevity. But I think if you frame it as reversing disease of aging, most physicians would realize that is their job, and there's nothing particularly alarming about it.
I'm in a humanities faculty, and even framing it in that way gets me responses of, no, this is probably not something we should be doing. So perhaps it's just where I am.
We should not be curing diabetes, cancer, heart disease.
Cure all that, but not aging or anything.
But all of those are age-related diseases.
I understand that. I have a difficult time transmitting that knowledge.
Just tell them that you're dealing with age-related diseases. That's what we do when we talk to the FDA. If you can hit multiple age-related diseases, you probably are hitting aging, but it doesn't really matter for the approval process, which is the key stage. Once there's a lot of people using it in its approved form for dealing with cancer, heart disease, stroke, diabetes, all these so-called common but late onset diseases, then I think it'll be de-risked. It'll be more acceptable. Many things that are unacceptable to begin. Most technologies are correctly rejected by a fraction of the population because they're unknown. Railroad trains were rejected as soon as the first accident occurred because they were dangerous, and they were. But that didn't take long to get the railroads back, and the same thing happens with in vitro fertilization, and almost in many biotechnologies, they're rejected until they're accepted.
So that would be actually the follow-up question, if you have just one more moment, which would be, what do you think the critical proof case would be in order to change that bulk of public perception? Like Aubrey de Grey has his mouse regeneration.
I don't think that influences people that have serious concerns. And animal models barely influence the FDA. I mean, they're just a necessary thing that you go through. I think it varies with the ethical problem or the safety concerns. With in vitro fertilization, it was remarkable how much resistance there was, considering there was no theoretical reason to expect it would cause great damage, and it was also surprisingly easily reversed by just N of one child, Louise Brown, in 1978, was enough. Neither should have occurred. There shouldn't have been bias against it, and there shouldn't have been bias for it based on one experiment. But in any case, now that 5 million children have been born with IVF, and for the most part, there are a few people that still oppose it, but it's adequately accepted worldwide. So I think that's the path that it will take. It's hard to tell whether it will be an N of one study or something bigger than that, but it'll probably go through the same FDA approval process that every drug does, and every drug is guilty until proven innocent, as it should be.
Thank you.
We have Dan, who hopefully his mic will work.
Okay, first of all, can you hear me?
A little quiet, but yes.
Is it better now?
Yes.
Okay, hello, and thank you. First of all, excuse my lack of knowledge. I'm just a hobbyist. I read a lot about biology and aging. My first question is, I learned about methylation of DNA, which I understood it's something programmed from birth, and which genes to be expressed, which genes to be shut down, and when. And I think that it's somehow interesting if it's possible to hack. Is it possible to hack this mechanism and to make it work as we want to work? Is it something that is studied and being researched?
Yes, so methylation is, yeah, that's a good question. The methylation is part of a interconnected set of levers that one can adjust medically and with synthetic biology It includes transcription factors and all sorts of epigenetics, as it's called. Methylation is a particularly good way of reading out aging. It probably has the tightest correlation with aging, on the order of, you can predict someone's age to within 2 years with assuming they're of normal
like
physiology.
Clock.
But you can also adjust epigenetics, which we've done. You can make any aged. You can go back and forth in time at the cellular level and the tissue level. So methylation is useful, and we do adjust it either directly or indirectly.
So is it possible in the future to just control it and tell him which genes to express and when, and just
yes, we can already control it. The challenge is delivering that control to every cell in your body or to specific cells in your body. The control mechanism itself works very well in the laboratory where you have access to the cells. It's a challenge to deliver it or any other gene therapy. But all of these technologies are improving very quickly, exponentially. So it's a little hard to predict how long it will take.
I see. Thank you. I will be very fast. Speaking of this, let's think about, I don't know, 10 years from now, 20 years from now, when a breakthrough will come here in the genetics of aging. How do you think it will be? It will be just a small, unique intervention that can be delivered through a virus or I don't know, or something, or it will be a mix of a lot of complex interventions that should be coordinated together. So let's talk about, I don't know, the future here. It will be
I think it's almost certainly a mixture. So if you look at the most powerful medicines for infectious disease and cancer, they're often a mixture of 3 different therapies. I think aging is sufficiently complicated that it's naive to think that it's one simple magic silver bullet. But it may be delivered in a very simple manner, and it will appear to be a single therapy, and it may even be a once in your lifetime therapy. We don't know those kind of details, but my guess is that at its core will be a mixture. Hopefully a simple and general mixture.
So apart from making sport and keeping myself healthy, I should just start making some money, I guess, for the future. Just a joke.
Well, hopefully we can bring the cost down. It should, if it affects everybody, it should be one of the least expensive drugs in history, because it will be the costs will be amortized very quickly over a large number of people. But it has to be done very safely.
Are you optimistic about this happening, like sometimes in our lifespan?
I'm not optimistic. I'm working on it. The best way to predict the future is to influence it.
Yes.
So I'm trying to accelerate that process, by recruiting, helping to recruit people and money and so forth.
Yeah. I donated for one research about the biological clock. I think it was like 2 years ago or something. Just a small amount.
That's definitely important work. The Horvath Lab and others are working on that clock.
Okay, thank you. Thank you very much.
All right, we're gonna go to we're gonna rotate around new people asking questions, and then if everybody gets through it, then we'll go back to secondary questions. So Joseph is next.
Yes, thank you. I have, depending on your answers, 4 questions. You do a lot of research with your own cells. Have you experienced that DNA information loss has been a major factor in aging?
DNA information loss is, I think, fairly rare. The main place that occurs is in cancer, loss of heterozygosity, deletion of tumor suppressor genes. And since they proliferate, it looks like a larger factor than it is at the core level.
Is that just because you observe cancer a lot, or can it happen silently?
It is because it proliferates. You get more copies of the cells that deleted. But there are ways that you can prevent that. There are ways that you can make backup copies of the.
Yeah.
You already have 2 copies of every gene. Most people do, and you could have 3 or 4. That's one way of protecting yourself.
Would you advise people against storing their own stem cells, like with embryos, for use at a later point in time for research that hasn't yet been completed?
I don't think there's any particular harm in storing your cells. They can be useful. They might be useful later if for example, if many cells in your body get contaminated by metastatic cancer, it's nice to have some that are back closer to the original state. But I think it's neither particularly good nor particularly bad idea. Probably the storing of cord blood was popular for a while. I think now it's more likely that what will be popular will be storing of adult stem cells, and if nothing else, it'll allow you a rapid response. So it's probably a net positive.
Mm-hmm. What, in your opinion, is a bottleneck in reducing the price of whole genome sequencing?
Well, the price of whole genome sequencing is now somewhere between 300 and 100. 300 now on the open market. It'll probably be 100 with the end of the year, a year from now. It's not clear you need to reduce it much more than that, because that's low enough so that the industry could subsidize everybody at $0. It's already the case that it's probably paid for itself 10 times over. The main problem is getting people to adopt it, getting both the insurance companies to be the good guys and give everybody a free genome and not look at it, and for people to see its benefit for serious genetic diseases. They tend to think of it as something that's less predictable, more frivolous, when in fact its main use is, I think, is for serious genetic diseases that you want to prevent happening in the next generation.
On that note, in regards to genome sequencing, how many times coverage do you think is necessary before it becomes useful for diagnostic?
Well, there's some utility at every stage, but I would say at 30x you start getting a fairly accurate representation of both your information from your mother and your father, which is important for determining whether you are have two copies of recessive or not, for example, among other things.
Is there a particular company you'd recommend for private individuals?
There aren't that many doing it for private individuals directly. Nebula Genomics is one that has put a special effort into making it highly private if you choose it to be, meaning encrypted, homomorphic encryption and blockchain and so forth. And they have the lowest price point, which is $300. So both from a privacy and cost standpoint and quality, they all have fairly good quality at this point. And you can always get a second opinion once you have the data. You can get additional interpretation. So I think it's mostly cost and privacy right now.
Okay, last question. Do you have a planned research or method for replacing or repairing neurons? And which of those is better, in your opinion?
I would say repairing is generally better because your memories are stored in the connections. And so if you allow the neuron to die and then replace it with a baby neuronal stem cell or progenitor, it has to make all those connections, and it has no particular way of doing that. We are working on ways that you can fix them in situ without changing their synapses, both either gene therapy or others. They're all some kind of form of gene or nucleus therapy.
Right. Look forward to it. Thank you, Professor.
Yeah.
All right, we have Lan next. Lan Lee.
Thank you, Professor Church, for giving us this question and answer session. I wanted to ask, could you talk a little bit about other factors or pathways you think are promising outside the work you're doing right now at Rejuvenate Bio?
Oh, right. So Rejuvenate is specifically looking for so-called non-cell autonomous factors. These are things that goes outside of the cell and affects other cells nearby or through the blood. So the other promising route is those that are cell autonomous, that stay within the cell. The problem with the ones that stay within the cell, like telomerase and the OSKM reprogramming factors, is that you have to deliver them to every cell in the body, or a very large number of cells, while the non-cell autonomous ones will diffuse away from the cell, and they can affect by transport processes the entire body. So I think we need both. To get the second category, we need better delivery, so we're working on researching delivery mechanisms.
Thank you.
All right, next we have Alexandru Voda.
Hi, can you hear me? So thank you for your talk, for answering questions. Just wanted to ask, I feel like most age-related diseases seem to be very polygenic and influenced by a lot of environmental factors as well. And given the complexity of the pathways involved and just the sheer numbers of interactions between gene activation, transcription levels, and so on, do you think the future individual constructs in a gene therapy might have on and off switches, inducers, repressors, or will we always use the existing natural promoter motifs to regulate that in therapies? Not just in research, because I know, in research, there isn't just about gene therapy, but, and does Rejuvenate Bio think of pursuing switchable gene therapies or something similar to that?
So let me just dissect that out a little bit, separate, because there's more than one question buried in there. So the first one is about polygenic, multigenic risk. Just the fact that there might be 1,000 genes involved, each of them having a very small effect, tends to flummox some of our colleagues. They just feel like that this is hard to diagnose, hard to do therapy on. But in medical practice, however, most things are polygenic and nevertheless are dealt as if they had a single cure. So, for example, height is probably one of the most well-studied polygenic traits. Thousands of alleles, maybe 9,000 different alleles explain 30% of the variance in the population. Nevertheless, in medical settings, you can get a huge impact with one gene, somatotropin, delivered typically as a protein human growth hormone. And it has very profound effects. So, and I think that's going to be very commonly the case. The reason I think I mentioned to a previous question that there will be multiple factors in aging therapy is not because it's polygenic so much as that you need to have you don't need to adjust all of the levers, all 9,000 levers, but you probably need more than one. So it's somewhere in between. The second half of the sort of part of your question was well, you maybe use your words. What would you say?
Whether you think, given the variance between individuals and genotypes, that maybe you want some sort of switchable switches inside of each construct for each gene.
So I've championed genome sequencing, which is one of the key components of precision and personalized medicine. That said, I think some of the most powerful medicine is generic, which is, we age as a species. We're aged quite differently than the mice and the bowhead whales, and so there is a generic component to it. We have generic vaccines, like we eliminated smallpox virus, it's extinct. That is something that applies to every human being on the planet. Maybe a few of us were resistant to smallpox, but not enough to base a medicine on it. And I think that will tend to be true for aging as well. I mean, there will be specific serious genetic diseases we have to solve before we can get to the aging, but everybody will have probably very similar aging. It will be epigenetic, which includes the promoters and so forth, but it's a little hard to predict the exact cocktail of factors. Probably you will work with the existing promoters, but with transacting factors that act on them.
Fair enough. And just a very short question on bioscience and bioinformatics PhD students that want to make an impact in the aging field. What sort of employment advice would you give them?
Yeah, bioinformatics is a good choice. It's something that can be used in a variety of contexts. I would add AI and machine learning to that. That's not intrinsically part of bioinformatics yet, but it will be soon, and it's extremely powerful.
Thank you.
All right. Next we have Esk.
Hello.
Hello.
I'm curious what you think is going to be an important or interesting problem to tackle after aging reversal in any discipline.
Probably in parallel with, if we're amazingly successful at aging reversal, the thing that we're all going to be worrying about, not just worrying about for our grandchildren, but worrying about for ourselves, will be climate change. It changes it from a next generation problem to a this generation problem, and I think the solution is not tightening our belts and starving ourselves of energy. It's carbon sequestration, and that means proper use of ecosystems. In particular, Arctic ecosystems are a great place because there are very few people living there, and the carbon that's fixed tends to stay around for thousands of years. In contrast, the carbon that's fixed in the rainforests and farms. But that's a much longer, separate conversation.
Thank you.
Looks like next we have Matt.
Yeah, hey. I'm just a hobbyist, so no PhDs over here, just trying to read and catch up. So I'd love to thank you for taking the time to answer my question. When I kind of think about, all of the different approaches to longevity and reversing aging, I know, we got epigenetic factors like with Sinclair, there's senolytics, there's telomere things. It seems to me that this is really a software problem more than a hardware problem. I know that there are folks that are born with hardware problems in their DNA, such as like type 1 diabetes and things like that. But I'm wondering whether that statement, tackling the epigenetics, figuring out how to reprogram this to go to it feels like a strange solution to change hardware when, to your point, we have whales that live hundreds of years or trees that live thousands of years on the same hardware. I'm wondering, how does genetic engineering fit into that, in your opinion? Where do you think I'm mistaken? What am I missing? I'd love to just hear your thoughts kind of on that, because it does kind of feel like the reprogramming epigenetics, to your point around the Horvath clock and aging around, methyl groups and those sorts of things, seems like the right way, but I don't know. Maybe I don't know if I'm missing anything. So I'd just love to hear your thoughts on that.
Well, I don't think you're wrong. You know, obviously we're all missing something, but yeah, I mean, it's a complex phenomenon that we're getting good control over finally.
Totally. I agree. It's a very complex system. So you know, I think that's where I'm trying to, because, you know, if the body can live for a year when you're 18 years old to 19 years old, then I guess in my brain, that body at 75 or 175 years old still has the ability to live for that extra year, but it's a matter of how do we make it live for that year, which to me feels like software, right?
Analogies like software and hardware are imperfect, but they're useful.
Yeah.
I would say that the DNA is kind of like software. Epigenetics is kind of like how that software acts on the hardware to perform functions. You know, it's like how the software of your you know, computer might control the hardware of your air conditioner to make your environment more pleasant. And the same thing with your body. But those are just analogies. We're getting very good at all of these. The software, essentially, our body has them all interconnected in a way. The software is intimately involved in the manufacturing of the hardware, and the hardware is intimately involved in making the epigenetics, which is the regulatory component. So, but you're basically the devil's in the details. We have to get specific molecules to do specific things at specific times in specific tissues.
And
that's
helpful.
And the analogies break down what we need at protocols.
Yeah, I understand that. That's a fair answer. That seems to make sense from what I've been reading. I agree with that. The only other question I had was, do you think that there's any red herrings so far, or, you know, what are some things that looked promising initially but are now getting overridden by people going upstream? Or, you know, has there been any, in your opinion, are there any areas of kind of the longevity, anti-aging, reversing aging field that now seem to be becoming less and less important because other areas are becoming kind of overshadowing them or overarching them?
I don't know of any legitimate research directions that have greatly, you know, diminished in value. I mean, there always will be some charlatan that comes up with something completely bogus, and I'm not even going to bother to give an example.
No. Right.
But you know, the legitimate things like telomeres and caloric restriction and so forth, they're now being joined by other players, but and some are easier to deliver than others. But that's all a snapshot of where you know, of our current progress report. That each of these things will go up and down in terms of where they fit into the probably multi-component therapies of the future.
Makes sense. Thanks a lot. I appreciate it.
I think Anton, did you have a question?
So I am Anton from Systems Biology of Aging Group, and I actually have a question about how to deal with extracellular metrics, because I mean, things like collagen, elastin ratio, stiffness, and other things that have geometry. So I am curious about your opinion about the best strategies.
Best strategies for what?
For solving aging-related problems with ECM.
All right. So I think this falls in the category where you can have things that look like damage, but are easier to fix than, say, you know, if you lose an organ, you can get a transplant. That's damage that can be fixed. Damage I refer to certain parts of the brain. If you damage them, they're very hard to fix because you've permanently lost those memories. With extracellular matrix, we're not absolutely sure, but I would guess that it falls in the category. If we make the cells surrounding it youthful, they will do the details of the repair for you. You don't have to micromanage it. You don't have to go in there with your nano shovels and shovel out the ECM. The surrounding cells will turn it over and replace it with youthful ECM. But that's a guess. That's not proven.
Mm-hmm. And another thing that I wanted to ask, I mean, how to deal with combinatorial explosions when you have a lot of different genes and you have to like tweak them differently depending on geometry, depending on timing. And even though we have such means from synthetic biology, the effect of like having too many combinations.
I think this applies to all of engineering. I mean, you could say that you have too many combinations in building a car. You know, does the carburetor interact with the right front wheel? The nice thing about both cars and bodies, human bodies, is that they're somewhat modular, and so you can debug one system without necessarily impacting another. In both cases, there are some things that do interact, and some of the things are additive, where if you fix two interacting things, you can get additivity or synergy. And in the cases where there really is, they are interconnected and it's not additive, you just debug a more complicated problem. And there are just a huge number of very complicated interconnected systems that we have turned out to have relatively simple solutions. Like the one I gave was for height, but the same thing is true for, vaccines address a high variation in the population in terms of their immune capacity, and a very highly complex immune system. But it turns out that the body is set up to have powerful levers that do amazing things, and we're just taking advantage of that. Developmental biology, for example, if I want to make a neuron, I don't have to, I just flip a single transcription factor and have a neuron. If I want to take a fibroblast and turn it into an embryo-like structure, 4 transcription factors. So the system is already evolved to make changes with very simple inputs. There are exceptions, however. Good question.
Thank you.
We have a user who's on a bus right now and can't use their microphone. They want to ask if cells reprogrammed with the OSKM factors or similar factors can regain its cellular aging hallmarks again after a certain time.
Yes, so you can do complete reprogramming where you take an 80-year-old cell and turn it into a zero, an embryonic cell, and you can do partial reprogramming where it just gets a little bit more youthful. It retains its epigenetic state. It retains its differentiated state, but it's youthful by many criteria. And you'll be seeing a few papers come out on this soon. It's I think one of the more promising methods. Again, the problem is delivery, is getting the OSKM Yamanaka factors to all the cells you'd like them to be in.
So this is the end of the official time.
Yeah.
I'm not sure if you want to continue. You don't have to continue if you don't want to.
But we'd probably have to do it some other time because I have a 4 o'clock appointment. No, I'm sorry.
Okay.
I'm having
That's no problem.
a great time here, and hopefully there's some benefit to you and others. So.
Yep. And we appreciate you stopping by. And
anytime.
yeah.
Keep up the good work.
Thank you.
Take care.